Hello, and thank you for joining us for today's presentation, part of our Adjuvant Cell Analysis Global Conference. I'm Susie Valdez of LabRoot, and I'll be moderating for today's event. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, simply click on that Ask a Question box located on the far left of your screen. I now present today's speaker, Dr. Sachin Katyal, an assistant professor in the Department of Pharmacology and Therapeutics at the University of Manitoba and senior scientist at the Research Institute in Oncology and Hematology at Cancer Care, Manitoba, Canada. For a complete biography on Dr. Katyal, please visit the Biography tab at the top of your screen. Dr. Katyal, you may now begin your presentation. Welcome, sir.
Thank you very much for that kind introduction. Today, we're just going to go over some of our recent data where we've instituted some new high-throughput DNA damage analysis methodology, looking at synergistic drug combinations that we can employ to treat malignant brain tumors. So what we're looking at primarily are astrocytoma, which is in the frontal cortex of the brain, and medulloblastoma, which is a childhood cerebellar malignancy that's quite common actually in children. So in the central nervous system, these two tumors are considered very malignant. So medulloblastoma is an invasive cerebellar tumor, which is 10 times more common in children. And it is thought that they emanate from immature or embryonal progenitors. In contrast, glioblastoma multiforme, or astrocytoma as I referred to it, is a highly aggressive form of brain tumor. It accounts for 30% of all tumors of the CNS and 80% of all brain malignancies. And it's thought to arise from dysregulated astrocytes. So current therapies in both tumor types include often surgery followed by radiation and also chemotherapy. And these chemotherapies vary depending on the tumor type, medulloblastoma versus glioblastoma. Primarily in glioblastoma, temozolomide, which is a DNA alkylating agent, is used. And as a second line, topotecan is known to be used. In medulloblastoma, vincristine and cisplatin are often used. Now, medulloblastoma does have a better treatment rate compared to glioblastoma. 30 to 40% of patients, however, do die due to recurrence, whereas glioblastoma multiforme has a 5-year survival rate of only about 2%. One thing that is common amongst both tumors is that survivors suffer from neurocognitive defects due to the side effects of the drugs. And these defects are much more pronounced in children with medulloblastoma because those are the formative years, and those children who do survive will encounter those defects for the length of the rest of their life. However, recurrence is very high due to chemoradioresistant tumor cells. And one of the aspects that we're looking at is whether this resistance is due to an enhanced amount of repair capacity to resolve DNA damage due to chemotherapy. Now, this slide is a very busy one, but it's really meant to give an idea of the different types and the breadth of chemotherapy that are used in treating a variety of malignancies. And the main take-home message here is that in over 98% of chemotherapies, the goal is ultimately to affect DNA, to instill some sort of DNA damage and overwhelm the cell's capacity to repair such damage so as to induce cell death. And so to understand how chemotherapeutics work on DNA and how we have this effort of cells to repair DNA damage in order to institute cell survival, we need to understand the different types of DNA repair pathways that are engaged. And so the cell has distinct DNA repair pathways to repair specific types of DNA lesions. Now, we think of these pathways as being very linear, but there is a lot of crosstalk amongst them. But this is a more simplified view of how DNA damage occurs. And some of the most pronounced lesions that occur are DNA single-strand breaks, which engages the DNA single-strand break repair or base excision repair pathways. We can have bulky lesions, which engage the nucleoside excision repair pathway. And what's most commonly known or referred to are the double-strand breaks, which engages the double-strand break repair machinery. Two types of repair paths occur. You have non-homologous end joining, and more commonly known and quite a bit in the news the last 5-10 years is homologous recombination repair. And so DNA strand breaks induce initially a cellular DNA damage response, so we call it DDR response. And this is orchestrated most often by the ATM protein. And so the ATM kinase is a PI3-related kinase, which goes on to phosphorylate a number of downstream effectors. And those include P53, CHEK2, AKT, CAP1, and it's thought to involve over 1,100 different effectors that affect the cell cycle, that can affect survival, that ultimately affect DNA repair, but also affect metabolism. And so one of the ways we know when a DNA break is induced is we can use an antibody against gamma H2AX, which after immunostaining marks sites of DNA double-strand breaks. So it's one way that tells us where the break is occurring. But if you look at this cartoon, H2AX is also involved in the early parts of the DNA repair process. So these indicate sites of potential DNA repair. But on top of that, we can use biochemical techniques to tell us what types of downstream effects have been mediated by ATM. This includes looking at CHEK2 phosphorylation, or serine 15 phosphorylation, and P53. Furthermore, we can look at ATM-specific effects, primarily CAP1 phosphorylation, which tells us where how well or how much the ATM pathway is engaged. So we have in front of us a lot of biochemical and molecular techniques that can illustrate what types of DNA repair events are occurring. Now, once we get past that DNA damage response effect, ultimately the cell wants to repair. And so for simplicity's sake, we're going to be looking at primarily DNA double-strand break repair and single-strand break repair. And so as I mentioned, in the double-strand break repair pathways, we have two different modes. We have homologous recombination repair, which primarily occurs when DNA is undergoing replication. And then outside of that replicative phase, we have the non-homologous end joining, which involves end joining factors that bring together broken DNA and ultimately reseal it. To complement these two, we have DNA single-strand break repair. This is a PARP-driven process where PARP will sense that DNA single-strand break, or NIC, and will recruit the extra CC1 scaffolding protein, which brings along a number of end processing factors and ligases that ultimately resolve these breaks. Now, to give context of the occurrence of these types of breaks, it's very often thought that irradiation causes DNA double-strand breaks, and it does. However, the ratio of DNA single-strand breaks to double-strand breaks is 40-to-1 upon irradiation. So it's very important to understand the context of the damage that's being induced and the different types of breaks that are being induced. But in the context of chemotherapy, and with the advent now of different DNA damage repair inhibitors, there's a thought that we may be able to improve chemotherapy success and possibly use DNA damage repair inhibitors as a way to chemosensitize tumors. And so for illustrating this purpose, I'm giving you a schematic of topoisomerase 1 inhibitors. So topoisomerase 1 is involved in changing DNA topology during DNA transcription or replication. And so there are DNA topoisomerase inhibitors, camptothecins, such as topotecan, irinotecan, and also there are non-camptothecin moieties that are also involved. And as you can see in this slide, a number of DNA repair pathways, including single-strand break repair, double-strand break repair, including homologous recombination, are engaged depending on whether the DNA is undergoing transcription or undergoing replication. And so we can, if we were to inhibit some of those repair pathways, so this is down below, we're talking about DNA damage repair inhibitors, we may boost the effectiveness of some of these topoisomerase inhibitors or top one poisons. So the idea here is, can we find ways where we look at different chemotherapeutics and DNA damage repair inhibitors and look at rational combinations and find effective ways to measure readouts using techniques that are effectively measure DNA damage that occurs in the cell. Current techniques are cumbersome. They're actually quite old. A lot of them were developed in the 1980s. And there are very few newer techniques that can look at this type of combination. And one of the most common old school techniques, as I like to call it, is the alkali comet assay, which can measure DNA damage at a single cell level. We can look at endogenous DNA damage. We can also look at induction of DNA damage de novo, but we can also look at repair of DNA damage by sampling different time points following damage or time after damage and measure repair rates. Now, the reason why this is called a comet assay is because at a single cell level, we are looking for the extent of DNA in the comet tail. So this comet arises due to damaged DNA that once you apply an electrical current, those damaged fragments will migrate away from the nuclear DNA. So it will form a bit of a trail or a tail. And when you visualize these cells under a microscope, they appear like a comet. And so the more damage, the more damaged DNA in the tail, and we can actually measure how much DNA and the intensity and the length of DNA in that tail and come up with a value that gives us a relative amount of damage. And so we call that a tail moment. And again, it's a microscopy-based image analysis where we're looking at the percentage of DNA in the tail, multiply the distance between the means of the head and the tail. And so, as I say, the old school way is for every time point sample we're looking at in say, a 24-well plate, we'll harvest those cells, put them on a slide, we will lyse those cells, wash them, put them in a comet assay slide vessel, apply an electrical current. We will add a cyber green to counterstain the DNA and image them under the scope. And then we will manually count this DNA damage or these comets and score these and come up with a value. And there's some semi-automated software where we point our mouse and click on the cell and it gives us a readout. But as you can imagine, this can be very cumbersome and labor intensive. And to sit there and count hundreds of comets per slide, when you want to look at different combinations of chemotherapeutics and DNA damage repair inhibitors, both in single, double, triple, or multiple combinations, you can imagine how many tens, dozens, or hundreds of combinations one would require to count. And this is where some of the new techniques have come out, which I like to call high-throughput comet, using a 96-well based comet slide and some of the new technology and analysis capabilities, some of the biotech instrumentation that is available. And namely, we use the Citation 5, where we worked with biotech to develop criteria and analysis techniques to make a semi-automated comet analysis possible. And so using common assay imaging analysis via the Citation 5 and Gen 5 software allows us to have an improved way of imaging and analysis of multiple combinations of DNA damage repair inhibitors and chemotherapeutics. So we get the benefits of automated imaging, which includes using predefined laboratory definitions, which allows us to have higher throughput capabilities. But also we have built-in cellular analysis capabilities where we don't have to export and import images into Image J to do individual cell, cumbersome cell analysis. Analysis of all comments are captured in one image. It's objective, it's free of user bias and fatigue. What you see is what you get. The system does the analysis for you. You're not, what we say, searching for comments where you're looking to click. It's not dependent on the user, so it becomes non-biased. And so we have this capability now to do this novel high-throughput comet analysis. And so this is a image readout of a common assay slide where each individual well is represented in each little box there. And so as we encounter more DNA damage, you start to see a transition from a no damage where you have nice round nuclei or nucleoids as we call them in this case, to when you have a situation of higher DNA damage where you start seeing longer tails that can be imaged. And a lot of this analysis and effort was developed by two student members in my lab, Asha Sinha and Marina Masazar, but also a lot of work was put in by Brad Larson from Biotech, who really helped make some of this automated analysis happen. So initial proof of principle analysis allowed us to start doing quantitative image capturing. So again, using a topoisomerase inhibitor, camptothecin, we could start seeing an increase in induced DNA damage going from a lower concentration to a higher concentration. And so using 2.5 times objective and the GFP channel, we could develop image montages, stitch them together, and do a automated analysis. And so this analysis will is two parts. We're using a dual mask identification. The initial mask is to identify the comet head, so it delineates the head, and then the second mask will delineate the comet tail. And then in doing so, a system can calculate the amount of DNA in the tail and therefore come up with the tail moments. And if you look on the right-hand side, when we do the validation studies where we're comparing more manual methods versus the Gen 5 analysis, the comparison stats are quite tight and they agree very, very well. And so building our resource around this methodology, we've put together our suite where we've got our Citation 5 cell imaging system combined with our Gen 5 Image Prime software. And we've been able now to increase our high throughput capacity by using a BioSpa 8 automated incubator. And in this case, we're not really even using the incubator portion, but we're using the automated plate capacity so that we can switch between different slides without very much user interface needed, so we can increase our analysis. So if you're looking at 96 wells on that comet slide times 8, we're getting close to 800 analysis all at one time. We've combined this now with an EL406 washer dispenser. We've got a multi-flow FX multimode dispenser, and also a Hamilton Star liquid handling, which allows us to truly use a more automated approach to institute this high throughput comet platform. Combined, this can increase our N number. We can then increase multiple combinations and use different cell lines, conditions, and time points, which allows for a more thorough analysis of DNA repair responses in different models. And as you can imagine, generally, just to give you some context, 2436 comet slides done in a more traditional manner takes us about 2-3 days of hands-on time and finally analysis. Whereas now we can do 800 individual comet analysis in a matter of 6 to 8 hours. So you can see we've got a scenario where we can really boost our throughput and enhance our analysis. And so this also allows us to start doing things like cooperativity studies, which we previously could not do. So this is the Prevagen comet assay slide. It's a 96-well glass slide, which will carry our agarose and cell mixture as beads, and we do all of our lysis, wash steps, and electrophoresis on this slide. So you can imagine a scenario where you could have drug A and drug B. In this case, we're looking at a topoisomerase inhibitor, topotecan, combined with a PARP inhibitor, olaparib. And we can then, in this case, look at 64 different combinations using different drug concentrations to do this analysis. And in doing that comparison, looking at topotecan and olaparib, where you're co-treating cells, and these are glioblastoma cells, for 1 hour, we can start looking at synergy analysis, analyzing the drug responses both individually and in combination with different drug concentrations. And we can extend these to other drug classes. Topotecan is a second-line therapeutic used to treat glioblastoma in certain cases. Temozolomide is the drug of choice, the gold standard. And again, we can look at temozolomide treatment over a 24-hour period with and without different concentrations of olaparib, and start looking at synergy analysis. Even from a biochemical standpoint, we can use a more pure double-strand break-inducing agent, something called neocarcinostatin, and combined with olaparib, look at DNA damage responses. And so this allowed us now to ask specific questions more about comparisons between different CMS tumors and amongst different lines within those tumors. And the idea here is, can we find different combinations of DNA damage repair inhibitors that would indicate that which could be maybe possibly upregulated, that show us alternate DNA repair pathways that may promote resistance to DNA damaging therapeutic agents. So in using these different inhibitors and in combination with therapeutic agents, can we identify combinations that may better sensitize tumor cells to therapeutics? So we're using DNA repair and genotoxicity responses to evaluate combinations of different therapeutics, different DNA repair inhibitors in different CNS tumor types and different lines within common CNS tumors. And in this case, for this proof of concept study, we were comparing glioblastoma with medulloblastoma cell lines. And so this work initially was spearheaded by Marina in the lab. And so an initial analysis was just to compare two different medulloblastoma cell lines where we were not treating these cells and then treating them with camptothecin and just looking at our initial comet tail analysis. And so this is just a simple 4-treatment analysis looking at 2 different cell lines. And this is typically how you would look at analyzing your comet tail moments. You count your comet tails and you would then express them as an average or a mean comet tail moment on a graph, just to look at relative damage amongst the 2 lines. And as you can see here in our concentration of camptothecin being used, the D283 tumor line is less sensitive to the Dowie medulloblastoma tumor line. But now when we expand this and start including different DNA damage inhibitors, this is where the power of the methodology really comes through, where we can use camptothecin in combination with different DNA damage repair inhibitors, both in single or in double or even in triple, and start looking at unique damage repair responses. And you start seeing patterns developing, particularly with the PARP inhibitor, where in combination with camptothecin, we see a multifold induction of increased DNA damage in both the Dowie line and the D283 line. And if we start interrogating these individually, we can start seeing individual differences amongst the lines here. But we could take this further and look at different DNA damaging agents. So looking at an alkylating agent, methylmethane sulfonate, MMS, combined with, again, our different DNA damage repair inhibitors, the PARP inhibitor, the ATM inhibitor, and another related inhibitor to ATM is DNA PK. And in combination, we start seeing unique signatures coming through, strictly using DNA damage and DNA damage repair as a readout. Again, breaking these up amongst the two different lines, we start seeing differences amongst the lines where specific sensitivities are emerging with different combinations. And then teasing these responses out, we can start asking biological questions. How do different repair inhibitors affect cell survival, viability, sensitivity? And here is an instance where we're finding that PARP inhibitors are much more effective in sensitizing the Dowie medulloblastoma line compared to the D283. And in conjunction with a topoisomerase inhibitor, we find that a PARP inhibitor can sensitize Dowie cells by 5-fold and enhance that DNA damage-induced cell death effect, whereas in the D283, we find very little effect of olaparib on those cell types. And starting to then extend this further, looking at cellular viability analysis, comparing the two different lines, we show that the topotecan is much more able to sensitize Dowie cells than D283. And then again, that's greatly enhanced once you add DNA damage repair inhibitors in the Dowie compared to D283. Using the citation 5, we can also use particular protocols looking at nuclear area, which enable us to do cell viability assays or cell growth assays. Such techniques are likely to be used in presentations that will be following mine, both with Kirk McManus and Navjeet Chander. They're going to be talking about some of these techniques in their ensuing presentations. But in this case here, we're using the nuclear area assay to look at growth, and again, instituting using olaparib with topotecan, we can see clear growth differences amongst the two different cell lines. And again, taking this further to different analysis techniques, and in combining with our BioSpa 8, we can do this in a kinetic basis where the system can analyze growth over time using different concentrations and combinations of these drugs. And so further, we can institute cell death assays using different cell viability and cell death dyes, and again, using this in a kinetic basis. And so we were interested in looking at what's mediating this clear difference between the two medulloblastoma cell lines. And so we performed RNASeq analysis, doing a triplicate analysis amongst our Dowie and our D283 medulloblastoma cells, and then using a lens of DNA damage repair in our readout, we identified a number of DNA damage repair genes that were potentially up and down regulated amongst the different cell lines. And then taking this to the validation stage, we went directly to Western analysis, where we were able to validate 13 of these genes as being differentially induced. And again, the most sensitizing of the agents in terms of DNA damage repair inhibitors in Dowie compared to D283 was PARP. And what we identified was, despite no differences in PARP genetics in terms of deletions or mutations, we found about a 80% reduction in the PARP expression in DAOI, which could likely account for the differential effect of the PARP inhibitor in DAOI compared to D283. There's less PARP in DAOI, and therefore that drug has a greater effect on PARP compared to D283, which readily expresses ample amounts of PARP, which would mean that you would need more inhibitor to have any sort of effect. And so in our mind, we took a very simple concept, the comet assay. We used a high throughput analysis technique where we could start interrogating different DNA damage repair inhibitors and comparing that with different chemotherapy modes. And we identified a cellular effect, a difference with particular combinations, and took that through to a validation stage where we identified a key DNA repair protein that may be mediating this effect. Now, just switching gears, we're looking at an analogous assay, and I already mentioned the gamma H2X foci assay. Now, this assay uses a gamma H2X antibody, which marks different DNA breaks. Again, a very old school technique where every break that's marked in the nucleus, and they actually look like little aggregates, or we call them foci. Each foci represents a DNA double-strand break site. And so you can imagine a scenario where every nuclei that you are imaging and then ultimately counting can be very low throughput, where you're sitting there and counting each individual foci. And you can develop scripts where each individual cell can be counted in a more automated way. But again, it requires you to import and export these images to image J, and can be very time consuming sitting there and compiling different individual foci. And as you can imagine, you would want to count a number of cells per well, a number of treatments, a number of cell types. It can become very laborious to do such analysis. And so again, we're taking our biotech instrumentation, putting it to work for us. Again, working with Brad Larson and Asha and Marina in the lab, we came up with a automated high throughput method of analyzing gamma H2X foci. And this takes advantage again of the imaging capability of the Citation 5, combined with some very good reagents and ultimately some multi-mask analysis techniques to come up with our foci analysis. And by doing so, we were able to expand our ability to do this analysis to look at functional cooperativity. And this enabled us to understand the dynamics of DNA damage repair. Again, DNA double-strand break analysis, a double-strand break undergoes repair through two different modes, as I mentioned, non-homologous end joining and homologous recombination. And depending what stage of the cell cycle the cell is in when that damage is incurred and that repair is ultimately undergoing, different nodes of the cell cycle will predominate. And we can harness different markers for the cell cycle and also markers for HR or an NHEJ mediated repair to understand how repair is occurring in the cell. So in addition to gamma H2X analysis, which allows us to mark DNA double-strand breaks, we can use antibodies against RAD51, which will tell us when a cell is undergoing HR-mediated repair, or 53BP1, which allows us to understand when the cell is undergoing non-homologous end-joining mediated repair. But in addition, we can do cell cycle distribution analysis using nuclear intensity and also different cell cycle markers, again, to mask along with our different DNA damage repair antibodies to understand what type of repair and when in the cell cycle this occurs, this repair is undergoing. Again, then using nuclear staining methodology, we can combine it with cell viability analysis and also use an antibody against anti-single-stranded DNA, which is previously shown to mark sites undergoing, cells undergoing apoptosis. So we can combine all these different analytical tools to come up with a one-stop shop way of doing analysis of dynamics of DNA damage repair and cell death. And so working with Brad, we first tackled DNA, gamma H2AX foci analysis, and coming up with our multi-masking methodology. We can first put our object mask around the nucleus, and then our secondary mask using Gen 5 image prime allows us to put masks around all the labeled foci that are labeled following gamma H2AX antibody staining. And then our secondary independent analysis can determine foci number per image. And so then by taking foci number per image, we can then calculate the number of foci per nuclei. Now, this was our initial way of tackling this problem. But since then, enhancements to the masking protocol allow us to look at individualized foci per cell, where now we can start analyzing each cell, each individual number of foci per cell, and come out with a readout. And so you can imagine scenarios where you can have untreated cells and you'll only see maybe 1, 2, or 3 foci, which are typical in replicating cells. You'll have all those S-phase dependent gamma H2AX foci, which are indicative of different sites of replication. So you can also image low damage, medium damage, and high damage, and nothing special about that. As you increase your damage, your number of foci will go up. But you can also have situations where the damage is so high that there's so many sites that could be gamma H2AX positive. Those numbers start going up and up and up, where you get a situation of having pan-nuclear gamma H2AX foci, where the entire nucleus lights up. So the readouts we're interested are in the number of foci per cell. And so some very quick analyses, we can start doing combinatory analysis, looking again at drug A versus drug B, and start characterizing the average number of foci per cell. Or we can look at the percentage of cells with a certain number of foci per cell. So we always like to look at cells greater than 3 foci, because typically cells have always 1 to 3 S-phase dependent foci per cell. And so here's a proof of concept where as we increase our concentration of our PARP inhibitor, olaparib, along with our topoisomerase 1 inhibitor, topotecan, we start seeing increased percentage of cells with greater than 3 foci per cell. But again, we can also look at number of foci per cell. And so you'll always have percentage of cells that don't incur many gamma H2AX foci and percentage of cells that incur a great number. But here you see in our heat map, as we increase our PARP inhibitor and our topoisomerase 1 inhibitor in combination, we start seeing increased gamma H2AX foci as indicative by the darker squares in our heat map. And so, well, we took this another step further to provide a proof of principle. We started using DNA repair deficient lines. A very commonly used line is the MU59 glioblastoma cell line. MU59J being a DNA PK deficient line, so DNA PK, is involved in the non-homologous end joining process of DNA double-strand break repair, and the MOVE59K, which is a DNA PK proficient. And so when we use these cell lines and we started instituting the use of a topoisomerase inhibitor, first looking at gamma H2X as the readout, we could see in our deficient line, we start seeing less repair as indicated by gamma H2AX in our deficient compared to our proficient. But we see an increase in gamma H2X pannuclear foci, indicating so much more damage that's being unrepaired compared to our proficient. But very nice to see our validation here, where in our deficient, so non-homologous end joining deficient MOVE59J line, we start seeing a reduction in 53BP1 foci staining. So 53BP1 would be an indicator of non-homologous end joining activity, whereas we see an increase in RAD51 or HR-dependent activity being enhanced in the non-homologous end joining deficient line. When we look at the proficient, we see 53BP1 foci being predominant compared to RAD51, although RAD51 is there. And that is consistent with the fact that RAD51 dependent homologous recombination is only occurring when the cell DNA is undergoing replication, whereas the 53BP1 dependent foci, which is indicative of non-homologous end joining activity, occurs throughout the entire cell cycle and is more often more predominant as a repair choice. So again, proof of principle, this showing us that we are getting what we would expect in the types of DNA damage repair arms during when damage is incurred. Now, using a variety of nuclear analysis and cell cycle markers, we were able to even look at cell cycle distribution of when this damage is incurred. And so this is the same assay using the same readout, we were able to show the percentage of cells in different phases of the cell cycle. And again, consistent with what we thought we should see in the deficient line, we see a preponderance of increased cell damage resulting in an increase in SG2 phase of the deficient compared to the proficient. We use another damage type, neurodevelopment, which is again a much more pure DNA double-strand break inducer. Very similar readout. In the deficient, we see a preponderance in RAD51 HR-dependent foci, whereas in the proficient, we see an increase in 53BP1 foci. So now taking this back to using clinically relevant drugs in glioblastoma, we wanted to look at how these cells respond to temozolomide, which is a DNA alkylating agent, and then the second line topotecan. And so temozolomide induces a number of base modifications which engage either base excision repair or MGMT, which may ultimately feed back into base excision repair. Now again, base excision repair is a single strand break repair pathway. However, these cells being tumor cells and being highly replicative, unrepaired DNA single strand breaks, when they encounter a replication fork, will collapse into a DNA double strand break. And so again, looking at DNA double strand break repair is particularly topical. And we were interested in combining those two chemotherapeutics, again, with different types and classes of PARP inhibitors. And so PARP inhibitors are very well known in the homologous recombination sphere, looking at BRCA deficient breast cancer, ovarian cancer, endometrial cancer, and is now being used in clinical trials in lung and prostate and colorectal cancer. But we wanted to assay a number of different types of PARP inhibitors, including a more recently developed one that's called pamaparib, which is known to cross the blood-brain barrier. Using an image-based analysis on our citation 5, in U251 cells, we were able to validate that this pamaparib and these other PARP inhibitors are effective in inhibiting PARylation levels. So PARylation is a result of PARP activity, and by inhibiting PARP, you see reduction in PARylation. And PARylation is often used to activate base excision repair and other double-strand break repair enzymes and other cellular processes. So in our hands, we show by image-based analysis that PARylation is effectively inhibited with or without DNA damage with these PARP inhibitors. And so we took this then to our assay. Now this is where the power of the automated high-throughput gamma H2X analysis comes into play, where we can use different concentrations with different combinations of PARP inhibitors, looking at our, in this case, temozolomide, at different concentrations at different time points, and start looking at our average gamma H2X and foci that form. And as you can see, as we increase particular PARP concentrations and different classes of PARP, we start seeing an increase in gamma H2X foci forming. And we start seeing themes where particular types of PARP inhibitors are more effective at inducing damage compared to others. But what's very curious is when you look at the higher concentrations, that heat map, that signal actually goes lower, and you would expect it to go higher with increased damage. And that led us to think that perhaps with the gamma H2X foci, we are not picking up foci any longer. Perhaps we're picking up more pan-nuclear foci. And indeed, we do see an increase in pan-nuclear damage with those two, olaparib and pamaparib, which is BMN 673, again, showing for two particular PARP inhibitors being more effective at chemosensitizing temozolomide to glioblastoma cell lines. But despite the fact that we see this increase, we feel that there were still some foci or some cells not being accounted for. And again, this is where the power of the all-in-one approach to this cell analysis comes into play, because we can use our nuclear staining to also co-measure cell viability. And so in this case now, we're looking to see where do we see a reduction in the signal in the heat map, where we see reduced cell viability. And again, looking particularly at the BMN 673, as we increase our temozolomide concentration with BMN 673, we see reduced cell viability, consistent with an increase in DNA damage-induced foci and an increase in DNA damage, ultimately resulting in DNA damage-induced cell death. Similar thing with the use of our topoisomerase inhibitor. As we increase DNA damage, we see more and more black, so more gamma H2X foci forming, but at the highest levels of damage, we see light, and that light is accounted for by the fact that we're seeing more pan-nuclear damage. So we're seeing a greater amount of damage, more so than can be picked up with foci, and we see an increase in DNA-induced, damage-induced cell death. And we can apply cell synchronization in these analyses, where we start looking at instituting different PARP inhibitors with our different DNA damage types, and we start seeing differential effects upon cell synchronization with the amount of DNA damage that's incurred, looking at both gamma H2AX, pan gamma H2X, 53BP1 foci, and RAD51 foci, which is then indicating to us at different phases of the cell cycle what types of DNA damage repair arms are being used to repair that damage. And finally, we can also look at DNA damage as a measure of cell cycle. and amount of gamma H2AX that are forming at different stages of the cell cycle, so that we can see where that damage is being incurred and how much repair capacity is present at different stages of the cell cycle. So what this allows us now, and it enables, is for techniques to study functional cooperativity. And we can apply this all-in-one approach not only to just live cells, but just imagine in a situation where we can use patient-derived tumor microarrays, particularly, say, in breast cancer, where we use neoadjuvant therapy to reduce the tumor prior to surgical removal of the tumor. We could actually look at how well did neoadjuvant chemotherapy work in the tumor by using different cell cycle markers and gamma H2AX to decipher where in the cell cycle those tumor cells ended up being in and what types of damage were incurred. We can utilize such a high-throughput gamma H2AX analysis in drug screening, look at FDA drug repurposing. We can do combination analysis and even use it in drug discovery. Again, through a lens of DNA damage. Again, the chemotherapies mostly are DNA damage inducing. Again, we can use this all-in-one approach to look at gamma H2AX, DNA damage, and ultimate DNA damage induced cell death. We can do drug responsiveness studies and compare tumor versus normal cells. So we can look at effective dose and therapeutic windows, comparing how well damage induction occurs in tumor cells and how toxic they may be in normal cells. And ultimately, this type of technique and methodology could be used for both diagnostic purposes, but even prognostication of how well tumors are going to respond. You can imagine scenarios where we can take live tumor cells from patients and do a multi-combination drug analysis and see how well this drug combination is able to induce DNA damage and do DNA damage profiling. So I believe the development of these techniques really opens up a lot of doors for both basic science and clinically relevant science and even patient care. And I think the sky's the limit on using these old school techniques in a high throughput setting for future fundamental and clinical analysis. So with that, I'd like to acknowledge members of my lab, particularly Asha and Marina, who've been the driving force for instituting these novel methodologies in our basic science and clinically relevant research platforms, but also like to really give a hat off to Brad Larson, who really helped develop and work with us. He came to the lab, worked with us and our instruments to develop these techniques. We had a very good collaboration with Brad, had a number of technical publications with biotech on the biotech website. But on top of that, we are now proceeding with publication with these techniques and some of our data, some of which I showed that are unpublished, but more data that we're trying to get published in the coming months. Been a very good and fruitful collaboration. I'd also like to acknowledge our funders, and with that, I'll take any questions that may arise from my presentation. Thank you.
And thank you, Dr. Katyal, for that informative presentation. We will now start the Q&A portion of this webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's get started. We already have some questions coming in, Dr. Katyal.
So I see one here. Can cannabis, THC, CBD can be used to treat malignancies? It's actually a very interesting question. We actually have a project in the lab looking at THC, CBD treatment in breast cancer, because breast cancer patients are amongst the highest users of THC and CBD amongst all cancer patients. And primarily that use is more for alleviation of symptoms and side effect management. But a fundamental question that has emerged is, can THC or CBD cause or change the tumor itself? Can it enhance DNA damage was the question we asked. And our preliminary data is actually showing, again, using these very techniques that I just presented, that so far pure THC, pure CBD, or combinations of whether 20 to 1, 10 to 1, 1 to 1, 1 to 10, or 1 to 20 THC, CBD does not seem to induce any additional DNA damage over normal.
Thank you, Dr. Katyal. And Dr. Katyal, is it possible to expand the HT-COMET assay to 384 well format?
We would love to be able to do that. As I mentioned, as I showed, there was a one clear in red slide. That's a Trevigen COMET slide, and that slide has a especially made where you have an image-ready surface to hold our cells that are mixed with agarose, and that those cells, agarose, are mixed together and they dry on that clear area of the slide. And then our lysis, wash, and electrophoresis occur on that slide along with the imaging. Our limitation is just on that slide itself, that we only have a 96 well type slide available. And so Trevigen makes that, and as far as I know, they have not made a 384 well format. So you could take a 384 well plate and then basically plate 4 different 96 well slides and then proceed from there. If you've got, a Hamilton or even some other types of automated liquid handling capabilities, it's not that difficult to do. You just need to have a few more manifolds to run the 96 well Trevigen slides.
Thank you, Dr. Katyal, and I want to thank our audience for these great questions. We have time for 2 more questions. What is the optimal concentration of cells to add to each well for the HT-COMET assays to get robust analysis?
So you have to be very careful you don't use too little or too many. Too little, you're not going to get enough N number. Too many, the comments are going to start to overlap and it's going to give the system trouble to do the individual math. So your goal is often to try to get between 20 to 30,000 cells per 100 microliters on each well. That's your optimal concentration. And you can find that those concentrations are specified in different COMET assay protocols, including the ones we've published, both with biotech and in some of my previous papers.
Thank you for that. And is the gamma H2AX image analysis assay you described available for download and use from biotech?
Yes, so it's on the biotech website. Brad and I have published a tech app note on the biotech website, and the more detail about both the technique and the analysis and the provisions of the assay are in that tech application note on the biotech website. So it's a very good resource to find more details on how to use that for that technique, and even the COMET assay, the high-throughput COMET assay, for your individual applications.
Dr. Katyal, thank you again for your presentation today. Would you like to provide our audience members with any final comments before we close? Again, we're looking at a very old school technique, but new uses in a high throughput manner. I think it's very powerful because it opens new doors to combinatorial DNA damage analysis, where we can now use these techniques for a variety of both basic science or clinical research. And I encourage you to explore this possibility, at looking at DNA damage and chemotherapy. And thank you again, Dr. Sachin Katyal, for sharing your work with us today. And thank you to all of our audience members for your questions and participation. Take care. Bye-bye.
Bye.